Hi, everyone. We're going to get started in a couple of minutes. Just going to give it a few more minutes for people to join. Thank you. Okay, we're going to go ahead and get started. Good afternoon, everyone. Thank you for joining the NINDS Biomarker Q&A webinar. My name is Rakanda Metley. We're going to start off with a presentation of slides, followed by questions and answers. Please submit your questions in the Q&A section featured below, and they will be answered at the end of the presentation. We have a full house today, so we ask that you please remain muted and keep your videos off at all times. I will be sending the link along with the presentation slides to everyone that has registered. This is being recorded. I will now turn it over to our moderator for today, Dr. Carol Taylor-Bird.
Thank you, Rakanda, and thanks everybody for joining the webinar today. As Rakanda said, we do have a full house, so looking forward to the discussion at the end. This is the NINDS Biomarker Program Technical Assistance webinar. I'm Carol Taylor-Bird. I'm the Program Director in the Division of Translational Research at NINDS for the Program Director for our Translational Biomarker Program. Dr. Ram Adrachandran is also a Program Director in our team, and he oversees the HEAL Initiative and Pain Biomarker Program. We also have Dr. Adisa Silu, who's the Health Program Specialist with us. And then, of course, Rakanda Medley, who you just met as Operations Coordinator. And we'll be talking today with all of you. So just quickly for the agenda for today, I'm going to go over a little bit of background and context for the NINDS Translational Biomarker Program. Some of you may be familiar with this aspect. Others, I know there's quite a few new people who have registered. I'm going to go over some important terminology and concepts. I'm going to be talking about the Translational Biomarker Program funding opportunities, including the analytical and clinical validation PARs that have been reissued, and they should be out on the street soon. And there are a couple of changes and new requirements to the analytical and clinical validation PARs, so I'm going to briefly touch on those. We're going to have a quick reminder about the NIH data sharing policy, and then, as Rakanda said, we're going to do the Q&A. So the Translational Biomarker Program at NINDS was developed and initiated to facilitate the validation of biomarkers and biomarker signatures as fit-for-purpose tools for use in clinical trials to accelerate therapeutic development and aid in decision-making for patient care. And you're going to hear me say fit for purpose probably 50 times in the next hour. So I think most of you are probably familiar with this definition, but just as a reminder, a biomarker is a defined characteristic that can be measured as an indicator of normal biological process, pathogenic process, or response to an exposure or intervention, including therapeutic interventions. And this is, of course, the definition from the NIH FDA Biomarker Working Group resource. So there's some terminology that we use in the NOFOs in addition to biomarker. We talk about biomarker signatures, where we're talking about a combination of multiple variables, multiple biomarkers, or composite biomarkers. Talk about detection method, which is basically any way that is being used to measure the biomarker, whether it's imaging or an algorithm or an assay, so on. Concept of interest, this comes right out of the FDA definitions, and this is an aspect of an individual's clinical or biological, physical or functional state or experience that the assessment is intended to indicate or reflect. So just as an example, I think most people are familiar with neurofilament light as a biomarker, and the concept in that case would be axonal injury or neuro degeneration. And then last year, FDA had, and NIH had put out a workshop looking at new terminology to address these multi-component biomarkers. And there's a link there that you can have access to if you haven't seen that. So just be aware that there's more terminology coming. So for thinking about applying to the NINDS translational biomarker funding opportunities, the first question you want to ask yourself is, what's the goal of your biomarker research? And we know that biomarkers, the term biomarkers gets used in a lot of different contexts in a lot of different ways. And then sometimes that's to think about understanding disease etiology or identifying new mechanisms. Or we're talking about using them as tools in therapeutic development or to use for decision-making in clinical practice. And so sort of the quick and dirty way to think about, should you apply to an R01 or should you apply to one of our funding opportunities? If you're really asking a disease mechanistic question, you'd go to the R01 or R21 space. If you're thinking about trying to develop a biomarker really as a tool, then you'd be coming into our program probably. Now, of course, there's a little bit more nuance here. This is an iterative process, and mechanism helps drive what tools are going to be appropriate in clinical trials. And sometimes those lead to better understanding of mechanisms. So recognize that there is some gray area. So as you may be aware, NINDS funds biomarker research across the translational spectrum. In general, a lot of the mechanistic work and some of the discovery work is funded through R01s, R21s, that's in our disease area portfolios in the Division of Neuroscience. We also have the development, analytical, and clinical validation space that the Division of Translational Research Biomarker Program facilitates. And then, of course, our Division of Clinical Research also has both implementation and some validation work in their funding opportunities in their networks. And of course, there's always an exception to a rule. There are also programs in our Division of Neuroscience that are really geared towards some validation or clinical trial readiness as well. But this is sort of to give you an overview of where you can kind of see the different areas of biomarker research. Of course, the small business program also facilitates biomarker research and device development and technology development. So why do we need biomarkers for therapeutic development in particular? We know that there's a really improved rate of success and improved clinical trial efficiency when biomarkers can be, robust and well-validated biomarkers can be used in clinical trials to advance therapeutic development. And it improves the rate of success at different stages, particularly between phase 2 and 3, and overall phase 1 through approval. So when are biomarkers used in therapeutic development? So this is just a cartoon to kind of illustrate some of the different ways biomarkers get used. In particular, pharmacodynamic response biomarkers are generally used in phase 1 and preclinical development, prognostic susceptibility risk biomarkers to help with patient stratification, targeting the right patients to the right trials, diagnostic likewise. Monitoring biomarkers can get used across the spectrum, and you can see some overlap between pharmacodynamic response biomarkers and monitoring biomarkers in some cases. Safety biomarkers help identify exposure, either to a therapeutic or to a toxin, et cetera. And so this is just to kind of highlight where they're used in therapeutic development translational spectrum. So just as a little bit more context about the translational biomarker program and why we have this, back in 2017, the FDA and NIH and public-private partnerships put out the paper on what evidence is needed for biomarker qualification. And I'm going to tweak the word from qualification to implementation, because we're going to talk about the FDA biomarker qualification program, but really the evidence expectations for biomarker qualification from the FDA are really kind of the same guiding principles as we think about for what evidence is needed to implement a biomarker, even if you're not going through the regulatory process. And in particular, where our funding opportunities are designed around is, and I'll just point out that, you know, it's for drug development, but we're talking about therapeutic development in any type of therapeutic. But our funding opportunities are really geared around the study design and specifically thinking about the context of use. And so what is the context of use? So this is a narrative that really typically includes the explanation of several components, both the purpose of how the biomarker is going to be used, when is it going to be used, what stage of development, how is the biomarker applied, and then what is the intended population. And so these elements are critical in thinking about your study design and what questions you're really asking in your research proposal for anything that you're applying to for our translational funding opportunities. I'm not going to go through all of these. This is the definitions of each of the categories of biomarkers as defined in the BEST glossary. Again, you're going to have access to these slides at the end. And so there are links to the BEST glossary here. I really want to get a particular point across, which is that when you're thinking about applying to the NINDS Translational Biomarker Program, besides the context of use being really critical, when you're thinking about how the biomarker is going to be used, you're thinking about what's the study design going to be. And so this is a publication that the FDA and others published in 2021, and it works through a couple of case examples for biomarkers that have been qualified relatively recently. And through the evidence review and statistical designs that were used for qualification. And in particular, I want to make the point that qualification, when you're thinking about applications to the translational biomarker program funding opportunities, you can kind of think of these first three steps as what would be in the application background, premise, and rationale, and the needs statement, and maybe the preliminary data supporting why this would be a biomarker that would be useful for a particular purpose. And then in these later stages, you're really thinking about the strategy and approach and the design of your study for the research question that you're trying to get at with the context of use that you're proposing. And in particular, I just want to highlight step 5 here, which is statistically quantify risks and benefits associated with using the biomarker for the intended context of use based on the totality of evidence demonstrating the practical relevance of the biomarker. So these funding opportunities are really geared to thinking about how are you going to know how to interpret the biomarker by the end of the validation process, and thinking about that from statistical and design questions. So as I mentioned, we have a series of funding opportunities from development through clinical validation, and I'm going to talk about those in a little bit more detail now. The development of biomarkers or biomarker signatures is our entry one. This is PAR 22089. This is an R61, R33 phased mechanism. And then we were hoping that the analytical and clinical validations would be published before this webinar happened, but they will be out very, very soon. I have posted the numbers here, so you know what to look out for. Don't ask me why they're not in order. That's not something we have control over. So in terms of scope, just sort of simply, in discovery and development, you can think about establishing that the biomarker reflects the concept of interest, developing or optimizing the detection method if needed, and then conducting proof of the initial clinical proof of concept validation study to test if the biomarker can be fit for purpose for one or more defined contexts of use, right? So that second phase in particular, you're really thinking about testing the premise of your biomarker utility. In the analytical validation stage, you've already have the evidence and can give the background and rationale for why this is going to be a fit for use biomarker. And now you're really evaluating the performance characteristics of how the biomarker is measured and thinking about things like sensitivity, specificity, accuracy, precision, and what kinds of variants there are in how the biomarker is actually being measured. And then finally, in clinical validation, you're establishing that the biomarker acceptably identifies, measures, or predicts the concept of interest within the defined context of use, right? And so key here is, are the results meaningful, and can you interpret how to use the biomarker results when it's measured at the individual or the participant or patient level? So very critical in terms of design. Qualification is not required in our biomarker funding opportunities, but again, if that's of interest, you're certainly encouraged to go that way, and that these funding opportunities can help support that effort. All right, so just in a little bit more detail, differences between the three sets of NOFOs in terms of preliminary data and premise. So in the biomarker development PAR, you're expected to have some preliminary data or supporting literature that really supports a strong biological rationale and premise for the candidate biomarker, or approach for identifying and verifying the candidate marker if you're not sure exactly what marker you're looking at yet, but the rationale needs to be there. You really need to have a strong supporting premise of why this is going to be useful and what the clinical need is. And you need to be specific in testing for one or more contexts of use. And you don't want to propose too many, because you have to actually test context of use that you're proposing that is going to be useful for. In the analytical validation PARs, you're expected to have already have evidence, whether you generated it or it's from the literature, about why or not the biomarker is going to be fit for purpose for a specific context of use. Again, supported by strong rationale. Clinical impact and unmet need is critical. How does this fit in the scope of other biomarkers or clinical outcome assessments that are available for the same purpose? And why would this be better? The detection method must have already been developed. All right, so you're not doing device development or assay development here. You've got to have a prototype already, but you can do some additional optimization. And then in the clinical validation, you're expected that the biomarker, again, you've already identified it, proof of concept for the context of use is there. Unmet need and impact is, of course, critical. And then the detection method must have already been developed, and there should be evidence that you already have a good standardized protocol with low variability that you can use how you're going to measure the biomarker appropriately. Now, differences in research scope for what you're actually proposing for each of the funding opportunities. In PR 22089, this is where the study design might include plans for preliminary clinical validation and are expected to include proof of validation, clinical proof of concept, to test the biomarker for a specific context of use. This doesn't have to be a multi-site study. It can be if it's necessary. It can include the development of the detection method, doesn't have to, but you do have to have that initial proof of concept study. In analytical validation, you can include optimization of the detection method, multi-site validation to see inter-site or inter-individual variability is expected but not required, although you do need to justify why if it's not multi-site. And then include the analytical validation of the methods, rigorous evaluation of the performance characteristics, any pre-analytical variables that might be confounding your interpretation, and establishing the standard operating procedures that would be ready to be implemented in a full-scale clinical validation effort. Finally, in the clinical validation PARs, you're going to establish the statistical thresholds needed to interpret and make decisions using the biomarker within the specified context of use. And we say context, you should really only have one, maybe two at most. More than that really isn't going to be feasible within a 5-year grant period. Large multi-site studies are expected. Recognize, of course, that there are some cases where there might be a small population, so you can justify why multi-site wouldn't be possible, but you do have to justify it. Clinical population should be large and diverse cohorts, and you need to really have sufficient representation for whatever the context of use you're proposing is. Plans to qualify biomarkers for the FDA is encouraged but not required. Some basic aspects to think about for each of these. So, of course, the mechanisms are different. For the analytical and clinical validations, we have both the academic U01s and the small business U44s. All of these are up to 5 years. In the R61, R33, they're phased. In the analytical and clinical, there are annual milestones, and we'll talk about those a little bit more. The budget is not limited, but it needs to reflect the scope of the project. For both the R61, R33, and the U01s, if you're going to exceed 500,000 direct costs in any 1 year, you do need prior approval to come in. We do expect bigger budgets in the clinical validation grants, but you do still need to do prior approval requests. Intellectual property, there's an element here that it's not scoreable, but you should think about it if there is an aspect that you're planning on commercializing. You don't have to do that. It's just something to be aware of and to include if you are planning to do so. Of course, resource and data sharing is required for all of them. A plan. So I more or less just said this, but for the phase mechanism, you can have 2 to 3 years for the R61 phase or 2 to 3 years for the R33 phase, but the maximum number of years is 5. And then again, reminders about the budget requirements and needing prior authorization. And that's at least 6 weeks before you submit the application, because it has to go through a internal review process. Just a reminder, make sure you include both phases of the application. If you don't have both phases, you'll be administratively withdrawn. Budget information here for the U44s, for the small business, and there's a link here to the small business budget information if you have more questions about that. And then again, reminder about budgets for the U01. All right, I'm going to give my voice just a quick break, and I think Adisa is going to talk about the next couple of slides.
Yeah, thank you, Carol. So as part of basically the research strategy section, we have to include, and grantees have to include timeline and proposed milestones for the funding period. And it is basically good to basically highlight, the importance of the milestones, and they should describe basically the project decision points, as well as have quantitative matrix for go, no go decision that will basically be utilized for the entire funding period. It is important to know that it is required to have annual quantitative milestones, and they should also have clear indicators of the project's progress, successes, as well as difficulties. And these milestones will be utilized in order to basically evaluate, the continued funding of the grants by basically the program official as well as the project scientists. As a milestone, it is important as well to provide a brief description of the success criteria, as well as basically the justification of the success criteria, because again, these will be utilized to basically evaluate the continued funding of the grant throughout basically the funding period. Next slide, please. Thank you. So there are in a sense two types of milestone, and it's actually very important to know the difference between the two. There are quantitative performance milestones and also progress milestones. And when we basically speak about the quantitative performance milestones, we basically want something that would define, for example, the target, sensitivities and precision thresholds that will be utilized for the detecting, biomarkers, which is basically relative to some of the, clinical gold standards. We want you to define, in a sense, some of the acceptable dynamic ranges, right, for the required context of use that is proposed in the grant. And also, for example, define any variation across the different sites or the operators, and also include, in a sense, the enrollment criteria and the sample, as well as the data collection goals for each performance milestones. When we speak about basically the progress milestones, we want to basically establish basically the contracts, as well as basically establish some of the standard operating procedures, as well as, the consulting basically with the FDA and the letter of intent. It is also important to include a Gantt chart when including that basically in the grant proposal. Thank you, Carol. We have basically important points when it comes to basically the not in scope studies that will not be, or that will be considered as non-responsive when it comes to any grants. And this basically includes anything that has to do with the development of a therapeutic agent as a primary intent, any evaluation, for example, of the clinical efficacy, again, as a primary intent, any natural history studies, any studies that are going to be using any animal models, as well as any studies that, fail to include those milestones, which is basically why we kind of highlight those as well. And it is important to note that anything that's outside of the NINDS mission will be considered as basically non-responsive. And with that particular point, anything that will be, non-responsive will be administratively withdrawn without review. Next slide, please. And I think I'll pass it on to Carol for the next slide.
I may have just frozen. Can you hear me?
Yes, we can hear you now.
Can you hear me?
We can hear you, Carol.
All right, sorry, I think my Wi-Fi cut out there for a second. All right, so thanks, Adisa. I lost the very last bit of that. So just for funding considerations, some reminders and just to be aware of. So applications within the top scoring meritorious range will be considered for funding. These are not percentile applications. So these are generally looked, we try to match the funding rate for the R1 or Payline driven institute. But within those top scoring range, we look at a variety of factors that are particularly important. So applications with the top scoring range need to address an unmet medical need, the significance, providing a strong justification for the utility and the need for the biomarker in the clinical setting or as a tool for clinical trial design. So, what you're looking at in terms of significance and innovation. Being supported by a strong biological rationale, the premise and significance. Including a carefully designed and a rigorous plan for performance evaluation. So that's your approach and milestones. And just to that point, the milestones are considered part of the approach and can be critiqued by reviewers as that aspect. Including a plan for standardization of data collection, samples for use and validation approach, and then have the expertise and resources needed for feasibility, which of course is the team, the available resources, as well as the approach.
Yep.
And then all investigators within the top scoring range are asked to respond to the summary statement critiques, and we go through a due diligence process with leadership for those top scoring applications. All right, so we're going to quickly talk about a couple of things that are specific to the analytical and clinical validation PARs specifically 2 new elements that will be required. And that's a team management plan that describes the workflow of the team and the key personnel. And this is required with a maximum of 3 pages. This was previously in the research strategy aspect. This is now a separate attachment to save room for your research design. And then plan for enhancing diverse perspectives, or a PDEP. And that's a required 1-page other attachment as well. And then this has been there before, but the associated clinical trial protocols and consent forms, if these are prospective studies or if you're leveraging ongoing clinical trials. So that's required if it's applicable, but not if it doesn't apply to your particular study. And then intellectual property plan, if applicable, again, depending on if you're planning on commercializing or not. And then, of course, letters of support as usual. So just a little bit more information with the team management plan. You really want to describe the workflow of the team and key personnel. You want to think about this being a multidisciplinary team, obviously for biomarker work as well as many research areas. You really need a lot of different expertise there, from the clinicians to the disease biology matter experts. Biostatistics, of course, is essential. Data management, data scientists might be particularly important. Technical experts with assay development, et cetera. And so we encourage you to form multidisciplinary teams, and I will point out, especially in the later stage validation ones, you can include consultants for regulatory aspects if needed. And then some elements to consider, and these are all outlined in the funding opportunities. And then also the new element is the plan for enhancing diverse perspectives. So the BRAIN initiative really led this effort, and I think you can probably expect to see this in more funding opportunities. We really want to encourage the research community to be thinking broadly about how to incorporate diverse perspectives to advance their scientific research and make sure that it's integral and equity and inclusion in the science that they perform. And so this document should include a summary of strategies to advance the scientific and technical merit of the project through expanded inclusivity. You need to include how it's going to be integrated throughout the research plan. You can incorporate this into the research strategy. And of course, it depends on the expertise and the project goals. There's a lot more information on the BRAIN initiative linked down here, and this is also in the NOFO. Here's some additional examples of that types of elements that would include inclusivity in research. I'm not going to read through all of these, but just to be aware of them, to look out for them and think about them. Finally, the data management sharing policy, as many of you probably are aware, NIH's new data management and sharing policy went into effect last year. So all applicants are required to include a plan. You're expected, you can include a budget for managing and sharing the data. This needs to be submitted in your application. This is reviewed by program, and you can discuss it if there's concerns and have additional requirements. But you really want to go through the new plan. It includes things like how the data are going to be shared, which data are going to be shared, where they'll be shared, when they'll be shared, and then, of course, associated costs. There is an enormous amount of information available, and I swear there's more coming every day on the NIH scientific data sharing plan. So here's a link to that and a few of the elements incorporated there. There's also a very useful FAQ on the data management and sharing plans. And there's a link here and some of the examples of types of questions that you can find answers to there. Reminder of a few of the resources that NINDS in particular uses or supports, including the Biomarkers and Biospecimen Data Repository, or BioSend. So if you're banking samples, you can do that through BioSend. You can also request access
What's going on?
Carol, can you hear us? Sorry, one, can you give us a couple of seconds? She may have lost Wi-Fi. Looks like she was completely dropped off. Adissa, do you know if that was the end of the presentation? Can we move on to questions?
I'm back.
There we go.
Where did you lose me?
You were just, finishing up talking about the repositories and resources, and then it got cut off.
All right. I apologize, everybody. I'm actually at the office and having internet connections issues. I'm going to share. Adisa, did you want to talk just briefly about?
Yeah, just briefly about the funding opportunity, if that's okay. Absolutely. So while Carol shares the screen, so basically the Translational Research Biomarker Programs has a new funding opportunity. And, for some of you that don't know, this is basically a program that funds any, biomarker signatures to predict the development of chronic pain from injury or various diseases. And, these characteristics can be used to define different pain levels, et cetera. But this particular funding opportunity kind of focuses on the validation as well as the development of remote or patient wearable devices for, again, to monitor pain as a use as endpoints in clinical trials. We have basically the receipt date that's coming up at the end of this month, January 30th, but we also have an additional receipt date, which is going to be on October 4th, 2024. If you guys are interested in this particular funding opportunity, feel free to reach out to me or Dr. Ram about it. Thank you.
Perfect. Thanks, Adisa. And then just some links that are in the slides that you'll be getting. And then with that, I'm going to stop sharing the slides and we're happy to answer any questions you have.
Thanks, Carol. Just a reminder, everyone, please submit your questions in the chat below and we will answer them for you. And again, for those that may have joined us later on, this has been recorded. We will be sending the presentation and the recording in the next couple of days. Thank you.
Thanks. Adisa, do you want to read the questions?
Yes, I was just about to. So the biomarker categories include, again, substitutability, risk biomarker, diagnostic biomarker, monitoring, etc. What biomarker takes priority when NIH funds biomarker research?
Yeah, thanks. So there isn't a prioritization based on the biomarker category. So, review is going to weigh in on the significance and impact. And so part of your application, when you're drafting it, is how is this biomarker going to be used? What's the unmet need? Why is this tool going to be useful for the field? And so that's really for you to justify and for review to comment on. And that's, a critical aspect of what your score will be.
Thank you, Carol. And the second question is, can we apply for these type of grants if we work on very rare disease where the sample size is really small?
Yes, you absolutely can. It is obviously challenging when you have smaller ends, and I know there are statistical approaches to get around that. So I would, reiterate and encourage you to work with your statisticians about thinking about how you're going to design your studies when you're dealing with small ends. But yes, absolutely, rare diseases are open.
Absolutely.
Maybe the next question.
So the next comment was basically a call for collaboration. But there is a next question, which states, in the early pain study, are behavioral therapists and occupational therapists able to apply? Or I think that might be maybe referencing to the HEAL
Oh, the
biomarker.
HEAL one. All right. Yeah, if that's for the HEAL one, then I'd encourage you to reach out to Ram and Adisa. And if you have a draft aims page, that's always helpful. And if we didn't get that correctly, then feel free to unmute. Thanks. Right, as we're waiting for others to think about any questions, of course, our contact information is available on the website and in the email that'll be sent out. So if you want to talk about a specific aims page or a project, we're always happy to have a call and talk about it and see if it's the best fit for this program or for a different funding opportunity. We didn't talk about digital biomarkers specifically. I see the collaboration, thinking about that. Currently in the funding opportunities, wearable-based digital biomarkers or functional assessments are in scope. So that's certainly open as well. And there's going to be some additional slides in the PowerPoint that will be sent out that includes some guidance documents to FDA documents. And so those are worth taking a look at if you're interested in going that route. The webinar, Rakonda, I guess I'll ask you, when will this go out?
Yeah, no later than Tuesday of next week. Yeah, Tuesday, because Monday is a federal holiday.
Great. Thanks, Rakonda.
No problem.
And one other thing I'll say, we do these webinars once or twice a year, usually a month or two before the application due date. The scope hasn't changed a lot. We try to tweak these a little bit to get, to be as clear as possible. But if anyone has content that they're really interested in or something that they were hoping to learn about on the webinar and didn't, I'm always welcome to get an email and we can incorporate it if possible in the future.
There is another question. Should preclinical genetic mouse models, I believe, always have to be accompanied with human data or orthogonal validation in a successful grant?
So I did not, it's in the non-responsive criteria, but I didn't highlight this point. So the development PAR and all of the validation PARs, animal work is not in scope anymore. So, if it's work supporting the premise and the biological rationale of why this marker is appropriate for important for the disease process or the etiology, then that's obviously part of your background in supporting literature, but these programs don't support animal work within the scope of the projects. Does that answer your question? Great, thanks. Yeah, and in particular, pharmacodynamic response biomarkers, when you're developing a therapeutic and thinking about what's a downstream target that you'd expect to see in response to your therapy, sometimes those are being done in your earlier preclinical work, whether it's an R1 space or one of our translational programs that helps with therapeutic development. If you're doing some validation work or looking, verifying that the biomarker is appropriate in human studies, then obviously those can come into our program. Any other questions? All right, and I don't think I specified the receipt date. So one thing, so for the development PAR, that's the R61, R33, the next application due date is February 22nd. NIH policy is that any funding opportunities, there has to be at least 60 days between when a funding opportunity is published and when the applications are due. So because the validation PARs are not out yet, you can expect you've got basically 2 months before the applications are due. We will send out the notification as soon as those are published to the biomarker listserv and the Division of Translational Research listserv as well. All right, give it 1 more minute, and if there aren't any other questions, then we can wrap up a little bit early. Yeah, thanks, Greg. We're glad to see the PDEC coming through too. I think we'll see more of those soon. All right, great. Thanks everybody for attending, and have a good evening.
Thank you all.